It's my pleasure to introduce our first speaker today. All right. Dr. Jenna Eckwaller is a biodiversity genomics postdoctoral fellow in the OCIO Data Science Lab right here at the Smithsonian Institution. She earned her PhD from the University of California, Berkeley in 2020. And her research focuses on the evolution and ecophysiology of dryland mosses. This afternoon, she'll tell us about the secret lives of desert moss. Jenna?
Thank you, Eric, and thank you all for being here. I'm really excited. Thanks to you for tuning in on Zoom as well. And of course, before I start, I would love to thank the organizing committee, everybody who worked to put this together, and for inviting me. It's my pleasure to talk to you about some of my favorite plants and some of their most exciting features. But before I get into the specifics of my work, I have to introduce mosses and point out to you that they're not like other plants in many ways. One way that's important for this work is their water relations are very different from most plants that you're probably familiar with. Mosses are poikilohydric, They're poikilohydric, which means that their cellular water content equilibrates to the external water environment. This is as opposed to homeohydry, which is most plants we're probably familiar with, plants that attempt to keep their internal water environment at an optimum, regardless of what the external water environment is like. So this means for mosses that live on land, they, when water isn't present, they dry out completely. Now, you probably have all seen this walking through a forest after a rain, or even a city like today, and the mosses have greened up and everything looks very lush. Well, this same thing happens in deserts. Mosses that live in the deserts dry out and green up after a rain. But in desert plants, it's pretty extreme. With those very low relative humidities you get in deserts, mosses dry out even more, survive extremely low water potentials, and come back to life, so to speak, when water is present. In fact, when they are dried out or desiccated, this process is known as desiccation tolerance, there's actually no metabolic activity whatsoever. So they're really, really quiescent is the right way to put it. The group I'll be talking the most about today is this genus of plants, Syntrichia. And Syntrichia has a worldwide distribution and occurs in a lot of different habitats, a lot of physiological and morphological variation. We have a whole project dedicated to studying that variation. But the group contains a lot of dryland specialists, a lot of species that live in deserts in particular, where they play really important roles in biological soil crusts. And in fact, this group contains the most desiccation-tolerant plants known of all plants. Like, for example, this Syntrichia caninervis. This video, you saw the plant was originally dry. I collected it from the Mojave Desert, and I sprayed some water on it. And you can see its leaves are opening up, and it's greening up before your eyes. And I'd like to stress that this is in real time, not a time lapse. It really is that fast, and it's amazing to see. They really do open up, turn green, and get to work. They start photosynthesizing and repairing immediately. So they're really good at this drying out thing that all mosses have some ability to do. And it was actually while out collecting samples of this plant, Syntrichia caninervis, for a population genetics study, when my collaborator and I kind of came across something unusual. We were in the western Mojave Desert, high elevation, just about at the pinyon juniper line where it starts changing to a forest. My collaborator, Kirsten Fisher, picked up this white quartz rock, and we saw there was moss underneath. And at first we thought this was like, oh, how funny, the rock covered the moss, and the moss didn't die. We looked a little closer, and the moss was still hydrated, actually, which was super unusual because it was June, and it hadn't rained in weeks, and this moss was sitting there fully hydrated. So we picked up more rocks and kept finding moss under these white quartz rocks in the area. So, like any scientist, I went to the literature, and I could find only one single report of mosses that live under rocks in the Mojave Desert, which would be called a hypolithic moss. And it was this paper published in the Bryologist in 1989. And it really wasn't much more than what you see here. It was truly just a report, a paragraph of a sighting of the moss Tortula inermis growing under a crystalline rock in the Mojave. I kept searching and found that there's a fair amount of research on microbial hypoliths, and in some of those papers there would be a note that mosses also occurred. So it exists, I knew that much, but I could not find any kind of quantification or characterization of this habitat, especially in the Mojave, as most of these were from Antarctica. There was also one cool species that's an obligate hypolith, endemic to the grasslands of Kansas, but that's all I could find. So my collaborator and I set out to characterize this habitat. We wanted to know the community composition, so we collected samples under the quartz rocks and next to it on the soil surface to compare. We measured and quantified light transmitting through the rock because they are photosynthesizers after all, and they need to get some light. And we deployed temperature and humidity loggers for 6 months, and recording every hour to characterize the microclimate. And again, on the soil surface and under the rock to compare. So doing moss community composition studies, you have a lot of data like this. You have to do microscopy and look at very small details, and these were the major players there. Syntrichia, caninervis, not surprising, this was what we were there to collect actually, and it's the dominant moss of the area for sure. A little more surprising was we found a fair amount of this Tortula inermis, the same one from that Bryologist paper at a completely different site. And this species normally doesn't occur here, or at least no one had really seen it because they weren't looking under the rocks. And this is what the plants look like more at a normal scale. There was also some Bryum argenteum, which is frequent in low abundance in the desert, so I don't have much to say about it. This is a visualization of the community there. The purple on the top is the soil surface, and the green is hypolithic, and then the three columns, Syntrichia, caninervis, Tortula inermis, and Bryum argenteum. So as you can see, the size of the box tells you kind of how many samples. Syntrichia caninervis was pretty abundant and more so on the surface, and the opposite is true for Tortula inermis, much more abundant under the quartz rock, and the Bryum argenteum was just found just once. So this Tortula inermis situation was surprising because this species is known to occur in lower elevations in the Mojave, which in the desert, lower means hotter and drier. And here we find it up the mountain, seems to be hiding out in these hypolithic spaces, which I first assumed would be cooler and a protection from the sun. So it didn't really make sense why we would find this plant apparently adapted to drier conditions and hotter conditions, kind of taking refuge at this elevation. So I looked at the temperature data to see if I could help answer that question. And on average, indeed, the daily high temperature under the quartz rock was 2 degrees Celsius cooler than the soil surface next to it. And the daily low temperature was 2 degrees warmer under the rock. So it didn't get as hot during the day, and it didn't get as cold at night, which both are important in desert habitats. But since I collected this data every hour for these months, I can do much better than these averages. So Here we've got the data as a line, and when the line is light blue, it's a daytime hour, and when it's dark blue, it's night. The y-axis there is the difference between surface and hypolithic temperatures. So when it's right at 0, there's no difference between them. And, when it's above that line, it's cooler under the rock, and when it's below, it's warmer under the rock. So as you can see that most of the time during the days, it's cooler under the rock, and at night, it's warmer under the rock. Except for these 2 periods where it levels out. Those actually represent 2 snowfall events, which this elevation in the Mojave does get snow, where there's no difference between day and night, but there's a slight but consistent warming effect by the quartz rock. It's slightly warmer under the quartz day and night during the snowfall events. So what this tells me, first of all, is there's a buffering effect happening, that it does, the extremes are dampened a bit with temperature, and also the restriction of Tortula inermis might not actually be so surprising, because if this plant is found normally at lower elevations where it's warmer, the rock actually provides a fair amount of protection from cold, it seems. And so it may reflect lower levels of cold tolerance in Tortula inermis, and that's why it's persisting under the rocks at this elevation. So it was it turned my assumptions on their head, because we think of deserts as hot places, and they often are, but they can also be very, very cold places. Another thing I noticed when looking at these plants is that Syntrichia caninervis, which occurred in both surface and hypolith, looked very different when they grew under the rocks. So these pictures here, the one on the right is a very typical soil surface Syntrichia caninervis. They're usually short shoots, dark brown coloration. And the one on the left is a pretty typical hypolithic Syntrichia caninervis, longer and greener. And indeed, I measured 349 shoots and found that they were 62% longer under the quartz, Syntrichia caninervis. And so you have to remember, with desiccation tolerant plants, they can only grow when they're hydrated. So if they're longer, they either grow faster or have more time wet in which they can grow. So this led me to ask whether under the hypoliths, the plants experience longer hydration periods. Now, I don't have data that can really get right at hydration periods, but I used relative humidity as a proxy, because if it's humid under there, the water is more likely to persist for longer. And again, we can look at the means. The mean daily low relative humidity is 30% more humid under the rock than it is on the soil surface. So we're getting our protection from that low drying point. There's no difference in the mean daily high, though. And again, we can look at the data over time. And we see a couple of patterns emerging. In these warmer months, in this part of the desert, kind of winter starts pretty late. Usually it's hot still in October and even November. And in the warmer months, there is a difference in relative humidity in a day-night cycle, like with temperature, where during the day it's more humid under the rock, and at night it's actually less humid under the rock. Remember, again, these are photosynthesizers, and so they can grow only when there's light, or they can photosynthesize at least only when there's light. So it's really important that they're hydrated during the day in particular. So that benefit there of being more humid under the rock during the day, that's key. And then in the colder months, it's actually, we don't really see the day-night cycle at all anymore. It's just a constant, almost constant benefit of being in the hypolith in terms of relative humidity. It's almost always more humid under the rock than on the soil surface. And again, this is in these plants, they're desiccated for much of the summer. And their growing season is in the winter. So this is key. This is when they need that humidity to grow and to thrive. So the quartz is really providing a benefit here. So from desiccation, which when they desiccate, they can't grow, they can't do anything. And in the deserts, desiccation can happen really quickly after a rainfall. So any extra time can be really important, which it appears to be based on their growth. But I keep mentioning this photosynthesis thing. They do need light, and these rocks, although apparently translucent, they look really white, they look opaque. So we wanted to know how much light was actually getting through. It turns out there was a group of geologists, Schlesinger et al. In 2003, that studied this very question on Mojave Desert milky quartz, this type, and developed an equation to calculate how much 650 nanometer light transmits through the rock as a function of thickness. So our average quartz thickness was about 2.5 centimeters, and for that thickness, it would be about 1.2% of light, of 650 nanometer light reaching the moss, which is pretty small. I also wanted to test this empirically, so I took just 2 rocks that kind of represented the range of thickness, and I measured total light intensity transmitting through. And as you can see, these 2 rocks, the top one, which is about the upper end, the thicker one, gets 0.4% of light transmitting, very, very small amount, but apparently enough. And the bottom is a 4%. So those are pretty consistent with that average 1.2 we're seeing. But there's another element to this that I thought that was really intriguing to me. This is from that paper, that geology paper, and the gray line on the bottom is light transmittance, and with the wavelength of light there. And it's slight, but you can see there's an incline in that line, in that gray line. And what that says is that bigger wavelengths pass through the quartz rock more easily than smaller ones. And that's important because the smaller wavelengths is where ultraviolet radiation is, and which can be quite damaging to plants, not always, but can be. And so they're not only getting really low light under the rock, they're getting different light quality. And whether that's good or bad may be yet to be determined, but it's distinct for sure, with a lower proportion of ultraviolet light. So that was the one story, but I wanted to talk to you about their life cycle too, because it's pretty fascinating. You'll have to talk to me more about the UV stuff, because I have a lot to say on that if you're interested. But another way that mosses are not like other plants is in their life cycle. Okay, like all land plants, they do the alternation of generations, but it looks a little different than most plants that probably we're all familiar with. For one, they are haploid dominant. So their main body form is, has just 1 copy of every chromosome. And in that, sex is expressed and determined by a single sex chromosome, U for female or V for male. Sexual reproduction then occurs by a flagellated sperm that reaches the egg to produce a sporophyte, which has both sex chromosomes and is unsexed, which then produces spores, each of which has either the U or V to then become a male or female gametophyte again. Asexual reproduction is common in mosses, and especially in desert mosses. Plants may or may not ever produce their sex organs. They can just grow as a sterile or non-expressing shoot, like this one on the bottom in that box there. And they can clone, which can occur by fragmentation, just literally rip off a piece and grow a new shoot. Specialized asexual propagules, such as gemmy, or by selfing. If they do make those sex organs, some of them can self. In desert mosses, and especially in Syntrichia caninervis, there's an extreme sex ratio bias with many more females than males. But when I say that, I'm talking about phenotypic females and males, those that are actually making their sex organs. You'll have at least 5 females for every 1 male, depending on the population. But as I mentioned, they don't always do this sexual reproduction, and many mosses never make their sex organs in this species, and so we don't actually know if they're male or female. So for example, take this hypothetical population of Syntrichia caninervis, and you look at it and maybe 80% of the shoots are non-expressing. We can zoom in on those expressing ones and see that there's a female bias, which here is represented as 7-to-1, pretty typical. But we don't know if kind of the population overall is actually biased or if it's just a bias in their sort of sex expression rates. So there's 2 kind of ends of the spectrum and possibilities. It could be that the population is completely balanced. There's the same number of males and females, but males express sex less frequently, and therefore we see an expressed bias. This is known as the shy male hypothesis. Males just don't make their sex organs as much. But it could also be that there's a bias that's through the whole population, and the non-expressing plants are also biased, and their males are actually rare. So I call this the rare male hypothesis. To test these 2 hypotheses, which are not mutually exclusive, it could be something in between, I went to a sampled population in the Spring Mountains of the more eastern Mojave Desert in Nevada, and used this new restriction fragment length polymorphism technique, which is just a technique that utilizes amplification of a gene on the sex chromosome, that U and V I talked about, that you can use to distinguish between males and females at the genotypic level, so they don't need to be making their sex organs. We developed this super recently, and we're able to apply it to an existing set of samples that were collected more than 20 years ago for a paper in American Journal of Botany that was published in 2000. So in that paper, they looked at phenotypic sex ratios, checked for this is Bowker et al., checked for gametangia or sex organs on 410 shoots, and found a 5-to-1 female to male sex ratio. But 91% of the shoots were non-expressing. So we took those non-expressing shoots, applied this genotypic sex assay, and found an 18-to-1 female to male ratio in the non-expressing shoots. So indeed, we found that males really are rare, in this population at least. So we're looking at something like this, right? Like the underlying sex ratio actually is female biased. But then the next question is why? What can cause this bias? Because I talked about this spore production after meiosis. After meiosis, there really ought to be a 1-to-1 ratio of males to females with that splitting of the U and V chromosome. But for whatever reason, we're seeing a sex ratio bias in the kind of adult populations. Well, there are 2 processes that I can think of that cause male rarity in these mosses. One possibility is faster clonal growth in females. So if females just make more shoots, then when you sample randomly, you're going to pick up more females that way. And in fact, in this species, laboratory experiments do find evidence that females grow faster, regenerate faster from fragments than males do. And so it really could be what's going on here. However, another possibility is that they regenerate at the same rate, they express sex at the same rate, but males die out more. Males just don't live as long or as well, or they die before germination even. They die somewhere in their life cycle. And actually a paper from 2017 with this population genetics one I referenced, we found evidence that males have lower genetic diversity, which is suggestive of actually more increased male mortality in the species. This was at a different population, so we don't know actually the answer to this kind of these legacy samples from Bowker et al. 2000, But we have evidence of both of these processes, and so far what we're learning is that kind of every population might have a different combination of these processes, and it's really seems to be a result of historical factors plus environmental factors, and so there's a lot more work to tease these apart. That was maybe a lot, so I'll summarize a bit here, and things that we learned and are some takeaways. Mojave Desert quartz rocks provide a distinct microhabitat for hypolithic mosses that has very low light and a lower proportion of ultraviolet radiation, has buffering from extreme temperatures and longer hydration periods. And hypolithic Syntrichia caninervis plants grow longer and greener than soil surface plants. We also found out that Syntrichia caninervis, at least at several populations that I referenced, is female biased, both phenotypically and genotypically, and males are not shy, at least in this population. But I would be happy to see more and find out if they are. With that, I would need to thank my funding sources. I most literally wouldn't be able to do all this without their generosity. I had a lot. So thank you to the funders, to my collaborators, of course, that I worked on with a few different projects here, including not least the student researchers that worked with me over the years, one of which who became an author on this recent paper. So yes, thank you to their contributions, and thank you all again. I'd be happy to take questions. Thanks. It doesn't sound, but the Zoom can't, yeah.
Zoombies down here. Is it working now? Okay. Mm-hmm. Thank you, Jenna.
Yeah, thanks.
Technical difficulties solved. We do have some time for questions if there's anything coming in online or anybody in the auditorium has a question.
Yeah, hi. We have one question online from Bruce. He asks, has this phenomenon been observed in cold desert systems?
So, I guess yes, probably the most it has been observed is in Antarctica, but it hasn't been characterized to my knowledge. It's only these kind of microbial community studies that sort of classify this one had moss or this didn't, because they're interested in other things. So yeah, I think it's been observed at least, but there's a lot more we need to know about it for sure.
Okay, we have time for another question, yeah.
Questions for me. Do plants, can they switch sexes during their life?
No, in this species, they are determined genetically for one or the other. I didn't sort of mention that there are some mosses that are monoicous. This is a dioicous moss, which is sort of the equivalent of monoecious and dioecious in angiosperms. And those we actually don't really know how their chromosomes work yet, but presumably they have both sex chromosomes, and some of those do switch sex throughout their life. Some of them present both at the same time or on different shoots. So it's kind of, but yes, there are definitely some that do that. And yeah, I've focused on dioicous so far, but I'm very interested in that question because how it relates to kind of the bigger evolutionary the history of sexual systems in mosses.
Questions are pouring in now. Joseph asks, how does the moss get under the rocks?
That's a good question. We don't know. I mean, with mosses, we kind of don't really know how they get anywhere. We know that the spores go way up into the atmosphere and can get all around the world. So they seem to be everywhere, and they just possibly, wherever they land, if it happens to be a good place. This was in a wash, I forgot to mention, and so there is a lot of movement over, kind of long time scales. And so it could be that there's spores sitting in the soil, and at some point or even they get washed underneath. We did see one sporophyte under a rock, so it could, no clue, we just know they're there a long time.
If you wanted to farm these mosses for some unknown reason, how would you design the little greenhouses? Would you use quartz, or maybe there's another material?
I've seen some kind of artificial versions of this with litter in the desert. Some people have left glass or something, and you can find, but the problem is that can cause an actual greenhouse effect, like you mentioned, where it heats it up during the day. So, and which isn't ideal for these mosses. They can be hot when they're dry, but not so much when they're wet. So I think quartz would be a good choice. In fact, we have seen some quartz that's too translucent, you might not get as much hypoliths, or if it's too opaque. So I really do think there's a perfect Goldilocks rock, and nature found it. So yeah, I think it'd be good. Thank you.